Well, our first speaker is, he's got a heck of a career. 15 years at Chesapeake Systems doing integrations for some of the biggest companies in the United States. Really great friend of mine, phenomenal individual, and he's going to be talking about data in the DNA. Nick Gold, please come up to stage and talk about your DNA and where it's been.
How can I possibly really top that? Hello, as Chris said, I was with a systems integrator called Chesapeake for 15 years. Had a little bit of a career change about 6 months ago. I'm now working for a Boston-based startup called Catalog. And indeed, we are storing digital information in DNA. So we're going to talk a little bit about that and why you all might be interested at SMPTE. So DNA has a lot of very interesting characteristics as a data technology. And the way I like to think of it is, if you came to planet Earth from some other realm and you looked at every element on the planet and you said, oh, we have to build data systems out of some of the molecules on this planet, which ones should we use? DNA would be a pretty obvious choice. It is nature's data molecule. It's been doing data storage for over 3 billion years on this planet. It's very dense. This is reason number one why we might want to use it for storage. In fact, hell, 8K might even require it from what I was learning this morning. So we're talking about 1 million times more density than the theoretical limits of magnetic and solid state. So that's in my hand holding an exabyte, holding a data center. And calculations are that the entirety of all digital information humanity has ever created in the back of a Honda hatchback is the physical volume. So you've got a huge volume savings in DNA. It's really tiny. It's very redundant. It's actually very easy to make many copies of DNA. There are laboratory techniques for this. So once you have data in DNA, we can make gobs of it. There's really almost no penalty for making exponentially larger amounts of this data storage medium. It's very persistent, and this kind of has several areas of applicability. We're very used to digital storage technologies that not only have a shelf life of maybe 5 to 30 years, if you're lucky, and having sold them a lot, I never told people to trust an LTO tape for 30 years. It's not very reasonable. But even more important is that these technologies grow obsolete practically more quickly than they actually fail on us. DNA is, as I said, been around for 3 billion years. It's something that's going to be essentially permanently relevant to us as a data technology. And that's a very interesting thing, because not only can DNA molecules last, and there are techniques to actually preserve them, they can last for hundreds, if not thousands of years, and they're always going to be the same molecules with the same characteristics. There's no outmoding DNA from that perspective. But the stuff on the right side of the slide is where we at Catalog are actually even more interested, especially more medium and long term. Because if you think about DNA in a living organism, it's not just a static data storage technology or information storage. It's something that's very active all the time. DNA is leading to genes being expressed and protein folding. Enzymes are interacting with DNA. Every time your cells replicate, and thus your DNA is replicated, a little enzyme crawls down the newly created DNA and checks for errors in transcription and actually can correct those errors. What we're really talking about is a form of biocomputing, and it's going on inside of all of us at every given moment. And there's some very interesting characteristics to the computational characteristics of this platform. For instance, with data, when we're doing searches for data, we have to index all of the data. It's a very computationally expensive problem. And then we get a rapid result when we enter a query and a bajillion pages in Google pop up. But that didn't come for free. There's indexing of every piece of information that has to occur. DNA is very interesting. We can kind of stick a proverbial magnet into a haystack with DNA, and we can design special DNA sequences that essentially will find their targets, and then we can actually extract them. So you can think of this as searching for patterns within very large data sets without having to do any indexing. And we think whether that's a text query or whether that's finding inference for a machine learning model that you've found, you can kind of get the same responsiveness whether it's a gigabyte of data that you're doing that query through, or a petabyte, or even maybe an exabyte at some point. So this computational and data analytics potential of the medium is very interesting. It's really the only in-storage computing technology, if you want to think about it that way. So the idea's been around for forever. This guy, well, not forever. Mikhail Neyman, this guy definitely would have been a SMPTE member, I think. He was a Soviet radio technologist and engineer. He actually came up with the idea, wouldn't this be a great, not only data platform in DNA, but he noticed that it's something that is being computed in nature, and he thought, someday this is going to be a computational platform. The first actual instance of artificially manipulating a DNA sequence to have some artificial sequence was actually, again, a fellow who would be friendly to the SMPTE crowd, Joe Davis, a very scientifically minded artist, and he stored this rune in a DNA sequence. He basically came up with a very basic mapping scheme, and his goal actually was to inject this DNA sequence into a bacteria that we would send on a space probe, and maybe aliens would find this someday. It was sort of his response to the Voyager Golden Record. I don't think they ever actually used it in that capacity. But the idea's been out there a little while. Now, it kind of went on hiatus for a while, and a lot of people in this room, heck, I didn't know this until maybe a year ago, it's now possible to synthesize artificial DNA molecules in a lab environment or even in a manufacturing environment. This is a purely chemical process. You basically build the DNA double helix spiral one base by base, and I'll show you a little bit about the process in a moment. And so for roughly a decade, a little bit more, people realized, well, heck, data's zeros and ones, essentially. DNA has these 4 letters. We can create a basic encoding scheme and actually essentially build DNA molecules that are encoding digital information. Microsoft has done a lot with this. Harvard's done a lot with this. Microsoft actually has an ongoing research project with the University of Washington. And this gives you an example of what some of these more rudimentary encoding schemes look like. And having been with a video integrator for many years, I'll say the word codec at the catalog office means something very different, because it literally does mean turning binary data digits into the 4 nucleotide bases of the DNA alphabet. So this is largely how it's been done. We got 4 letters. Maybe we just make them each worth 2 bits of data. So let's say this is the message I want to encode, and this kind of illustrates the chemical synthesis process for how they build these artificial DNA molecules. Again, these molecules never were in a living organism. They're technically organic, but they're not alive. They're never going to go back into something living. It's just DNA in a dish, basically. So OK, A is 0, 0. We're going to add that base. We're going to add another. Oh, we've got these other ones for the other pieces of the message. And this process is, again, a chemical one. It's a kind of a 4-step process. And the problem is that every one of these bases that you add, this 4-step chemical process, is essentially about a 30-second operation. A 2-bit per 30-second encoding speed is not that great, not that scalable, but this is what people were essentially limited to. Now, you can do a lot of them in parallel. You can make many copies of the same sequence, but think about it obviously for a moment. If you have to take arbitrary digital information and turn it into these 4 letters, and this is the process that you're limited to, you're going to have to basically add every bit one by one, and there's really very little getting around that 30 seconds per bit, because it could be any string of digits that you might need to encode. You have to just kind of do them one by one. That's the level of granularity you're working with. So this shows, and that's a logarithmic scale on the left. This shows some of the efforts, again, over roughly a decade of people starting to use these chemical synthesis techniques. Towards the upper right, that University of Washington Microsoft indicator there is representing the largest published encoding job using synthetic DNA molecules. It was about 200 megabytes. And this shows you some of the interesting dynamics at play of encoding megabits or even low numbers of megabytes into DNA sequences. If you wanted to use these processes, and any of us can go to a website from several of the firms that offer these DNA synthesis services. You can send them essentially a text file full of A's, T's, C's, and G's. They'll send you a test tube back. Did you know this? A lot of weird things going on in synthetic biology that the public is not quite well aware of. Now, they do check your background and make sure that there's nothing nefarious in the sequences. But the price, a terabyte using these types of services would cost $20 billion. And the estimate is that the Microsoft UW 200 megabyte encoding job was probably about $800,000 in street price and took at least several weeks to encode. So not very scalable. So we came up with a very orthogonal encoding scheme. I think you guys will appreciate as engineers the sort of lateral thinking that our founders had when they realized a different way of solving this problem. So if you think about it, we have this molecule. It has these 4 letters. We need to use it to create states of entropy which can capture information, right? That's how you store information. So what we actually came up with is a combinatorial approach, and I'll illustrate it a little bit more in a moment. But I want you to think about it like this. If the previous encoding scheme that a lot of the other groups have done is basically a couple of bits per one of the 4 bases, we attacked it very differently. In fact, we did say we're willing to give up some of the density, because we don't necessarily need today an exabyte in a sugar cube. There's a lot of orders of magnitude to play with here. And so what our guys came up with was a technique by which we can prefabricate, using those DNA synthesis services, essentially a library. You can think of them almost as DNA movable type sequences. There are a couple of dozen bases long, and we have an alphabet in our current encoding scheme. It's a very extensible scheme, but currently we use a little over 100 of these prefabricated sequences. And you might remember, we can order them in bulk, and by keeping them short lengths and having a standard set, we can kind of keep our costs down. So we go to those synthesizers of DNA as a supplier, something we might be able to do in-house at some point if we wanted to, but that becomes our building block. Now we've got these 100-some-odd short sequences. This gives us a combinatorial space of potentially trillions or even beyond of ways we can combine these prefabricated sequences into longer molecules that are in the ballpark of a few hundred bases long. So this is how we encode information into DNA. It's by rapidly combining prefabricated DNA sequences. In fact, we design our sequences so they have single-stranded overhangs. And you might remember, A always matches to T, C always matches to G. So we can design them so they will kind of line up almost like Lego bricks. We actually use an enzyme to fix them together more permanently into the longer molecules. And we call these combined longer DNA sequence molecules identifiers. And I'll explain to you exactly what an identifier is right now. So this is a graphical illustration. And I want you to think of those kind of colored chunks that you see in the upper left there as the shorter sequences that we might combine. It's not the exact number we use. This is just for illustrative purposes. We call them layers, but you can almost just think of them as orders of these shorter DNA sequences, and there's banks of them. There's a certain number of the prefabricated sequences that kind of represent possibilities for layer 1, or the first slot in one of these combined sequences. Layer 2, layer 3. As I mentioned, we have a few dozen layers typically that will combine together. So we actually know what the ordered position of all of these combined DNA molecules are. There's a specific molecule that we can generate that represents essentially slot 1, slot 2, slot 3, slot 487,689. We have very specific molecules that we create for all of these slots. So what do these slots do? And again, how does this encode an actual message? It's interesting, and this is something, frankly, that I thought was just very fascinating about how Catalog, again, kind of took a lateral approach to solving this. So let's say this is the message you want to encode, and again, it's either going to be a 0 or 1 for every slot of an encoding job. What we do is, if we want a particular slot, which you can think of as an address for a particular bit in the encode. So every one of these identifier molecules is essentially the address for one bit in the message. And so if we want to say slot 1 of this encoding job is a 1, well, in this case, I'll use what's on the screen, is a 0. We just don't create that molecule. And we know slot 1 is a 0 in that encoding job. We didn't create a molecule for slot 2. It's also a 0. Slot 3, oh, look, we sequenced it up. We found that molecule. That means for slot 3, we know that's a 1, and so on and so on. And so essentially, again, every bit has an address, and the identifier molecule that we've combined together is that address for that bit. So there's a lot of very interesting things we can do with this. So let's say we've created a bunch of these molecules, and I'll explain to you how we actually do that at rapid speeds in a couple minutes. OK, we need to figure out what's the message in this little petri dish of DNA molecules or a test tube, right? So we sequence it. We said, oh, we found these identifier sequences. We know what the position in the encoding job of every one of those molecules is representative of. We didn't find one for what slot 1 ought to be. We didn't find one for slot 2, or 0 and 1. We like to start at 0, of course. We know that those should be zeros. We found one for the position 2, the third position. We know that should be a 1 in the message. And this is really just how we do it. Every bit gets an identifier, or lack thereof if it's a 0. So again, we obviously gave up some density to do this, but it also got us around the problem of having to build every DNA molecule for an encoding job in a specific custom order, different for every encoding job we might ever encounter. So we can do a lot of things once we have these identifier molecules created. For instance, there is a laboratory technique. It's been around for decades. In fact, its inventor just passed away, called polymerase chain reaction. We call it PCR. PCR is a lab technique that allows you to amplify or exponentially multiply a set of DNA molecules using the polymerase enzyme. So we can, once we have a set of these molecules created, we can generate gobs of them. And that's actually very inexpensive. We can do it exponentially. It means, think about RAID 1, where you might have a couple of copies or something, or another kind of encoding scheme where you make multiple copies, maybe you have 4 or 5. We could have tens of thousands of copies of every bit. We could have hundreds of thousands of copies. We can split those into multiple containers, and you can have 10,000 copies of all of the messages in 100 different locations. It's very flexible. But we can also do random access, because the PCR technique has been adapted to allow you to target specific DNA sequences. So this is how we actually perform what's sort of equivalent to a random file access, although for us, it could be a file, or it could be essentially an arbitrary sequence of bits that are represented by these identifier molecules. So we can actually do this kind of hierarchically, and this allows us to perform random access of particular subsets of the overall data set. Essentially what we do is we overrepresent the ones that we're targeting. We overrepresent them through that PCR technique, and then it's really all about signal to noise. We see, oh, there's way more, orders of magnitude more of these molecules of certain portions of the message. Those are clearly the ones that we're looking for. So we've done a lot of testing. There's still much to be done, certainly. But we've done things like boil a sample set of these molecules for 4 days at 100 degrees Celsius, sequence it, found that there was no degradation in the message. And we're not saying we encourage people to boil their DNA data samples, but it shows you that these molecules are actually fairly stable. They're not storing a bit as charge on a gate. They're not storing a bit by the orientation of a magnetic particle. It is a physical molecule. It might be very tiny, but those bonds are relatively stable. And again, we have so many copies we can make, even if there are some errors at some points. PCR can introduce errors. We have so many copies, we can, again, sort of see what the signal and noise is in those. We can PCR a copy of this data set 1,000 times over, and again, get 100% accuracy in the message. Now, again, we have to do testing, but it gives us a lot of hope that this is a very scalable technology and one that can really store the data in a protected way for a long time. This shows a little bit about how we do sampling of subsections hierarchically in order to do essentially that random file access. So we have a component library. We need to kind of add these molecules together. We need to stitch them together with an enzyme. We actually have to incubate them at a higher temperature once they've been exposed to that enzyme, so the enzyme sort of seals those component molecules into the longer identifier molecule for that address. And then we have to pull it all together. Like, how do we do this at scale? When we first came up with this technique in the encoding scheme, we were using just standard laboratory liquid handling robotics, stuff you could picture in a wet lab environment. And it showed that the encoding technique worked, but this wasn't going to achieve the scale that we needed. We need to be able to do all of these things very quickly to do an encoding job. So what did we do? Well, we didn't intend to kind of keep up with the movable type printing metaphor, but we actually did end up using inkjet printer technology to do this. Because if you think about it, an inkjet printer nozzle and head, what does it do? It does very precise picoliter quantity liquid handling operations. And so we needed something that could very rapidly and very accurately combine the liquids that are our sort of DNA inks, and you see the actual ink in the upper left, that's the bank of the print heads that have the DNA inks that are those components that we add together. We roll this webbing through it, and I'll tell you a little bit more about the process in a moment. It goes through an incubator. We have these sheets that have these combined DNA molecules. We pull them all together. This thing is several meters by several meters. But this is the device that we call the Mobius, and it is the first generation custom DNA printer/writer that embodies our encoding technique. So on the right, you'll see that that's a little video. Those kind of orange, and the reason they're orange is because there's a dye that's used for quality control in the camera system that does QC. Those blocks are actually lots of those little dots that you see on the left there. But they appear as kind of rectangular blocks because they're just so small and so closely co-located to one another. You can see them going into the pooler after they've been through the incubator. They kind of fall into a basin of water. We actually can reduce down the water, so you're left with just a little speck at the tip of a test tube, and that could be gigabytes, terabytes. I mean, we're even talking petabytes in pencil lead volumes. It's very, very dense. So our first big encoding job, and we have lots of testing and optimization to do, but with Mobius several months ago, we did a 14 gig encoding job of the English text version of Wikipedia. And so that was bigger than the 200 megabytes that had been done previously. And again, the way we were able to do this is because of the encoding technique and the equipment that actually does the encoding. It was running at about roughly a 4 megabit a second write speed, not very fast compared to data storage technologies that we're used to. We think we can get this machine with optimizations closer to maybe 10 to 12 megabits a second, but we think that we have enough levers that we can push and pull and enough buttons that we can manipulate proverbially to take this fundamental technology in a future generation up to probably low gigabit per second write speeds. So now you're actually saying with a few of these things, we could start to capture significantly larger volumes of data into DNA. So this is an interesting quote from Richard Fineman. Fineman, obviously a great theoretical physicist, but also was very, he basically came up with the idea of nanotechnology. He really saw nature and molecular biology as essentially nanotech in action. And one of the things he observed about it, again, I go back to this, it's not just a data storage mechanism, it's a computing mechanism, is that these things in nature are very active. They're doing stuff. They are computing. Every cell in our body is computing at every moment. And there's a lot we can do with this. Certainly those types of rapid search queries without having to index is one of those things, but there's other types of data analytics that we think that this is a really ideal medium for. It could be finding complex inference of relationships in large and complicated graph databases. I want to see everything that's 8 edges away from one another and determine those relationships. We think DNA as a computational platform will be great for that. We've all been using von Neumann computers for our entire lives. This is the architecture where there's basically a processor, there's stuff that stores the bits, there's a bus that connects them, and a lot of people, maybe in this room realize, but in the wider world, that process of moving bits between storage and the processor is really one of the most limiting bottlenecks in computing as we've known it. What you can think of as DNA is the ultimately parallel computer, where essentially every bit of information stored in DNA can be molecularly manipulated simultaneously. So think it's almost like the opposite of a von Neumann architecture. There is latency in these types of chemical processes, but we think that the future is like, okay, we want to find this face in all of the video. If you can give us till tomorrow to do that, we think that that's something DNA would potentially be ideal for. So we are lining up pilot users. We have a few in the M&E space. I will say we are looking for a telco type customer because we think log data that is relatively small but grows to huge volumes and is stuff you might want to do specific queries against or you might want to perform data analytics against. We think telcos and the volume of that type of log data that they're generating could be an interesting pilot case. So if that's your realm, you're more than welcome to reach out to me. My email is nick@catalogdna.com. If you want to see more video of the machine in action and more explanation as to how it works, you can just actually go to YouTube and look for Catalog DNA. And I would be happy to take any questions about why we're doing this madness.
Thank you. Let's have some applause for Nick. What a mind-blowing presentation. And we do have 5 minutes for questions, yes. So.
Merrill, I knew you would have a question.
Actually, it's a comment. The comment is you came to the right place because SMPTE already has a standard that was evaluated against use of DNA storage.
AXF?
AXF.
We should talk, because one of my thoughts, and I've been talking to James Snyder about this, is the idea of maybe doing a pilot that embodies AXF captured information in DNA molecules. Again, our encoding scheme is different, but we can take arbitrary data, and frankly, for all the reasons that AXF as a self-contained, self-describing format is so interesting, if you are imagining molecules sitting on a shelf that maybe 1,000 years from now, someone might actually still be able to sequence and get data out of, AXF might be a really good way of presenting it out.
Yeah, for those in the room who don't know what AXF is, it's the archive exchange format.
Another thing I'll just mention is that the way we read data out of DNA, and let's say we don't want to just do analytics, we don't want to do queries, we just want to read the data back out. Catalog's encoding technique has completely turned the tables. A year ago, it was much cheaper and easier to sequence data out of DNA molecules in a relative sense than putting data into DNA. We've actually completely flipped the dynamic around at Catalog because of our technique in the Mobius writer. So reading is using standard sequencing equipment. Now, sequencers have improved faster than Moore's law over the last 20 years. They keep getting better. But it's interesting. They've been developed around having to read the granularity of each letter in the DNA alphabet, if you will, at that level of granularity, and that's what those sequencers are designed to do. If you think about it, the way we encode data, we have a different problem. We need to be able to tell all of these molecules apart from one another. It's a different type of problem, and because of that, we actually think that there are some newer, they're called nanopore sequencing technologies that we can adapt for our purposes, and we should be able to not necessarily get commensurate gains on the read side. Read is going to be behind write for some time, but we think we can actually improve sequencing technology a bit for our use case specifically.
And I think, Chris, you had a quick question too, didn't you?
I couldn't resist. Nick, a couple questions for you, because it's really interesting. DNA is tiny. How big is this machine going to be that has to read it? What's my data center looking like?
So the read side, there's many different types of sequencers. There's only several companies that really have staked down the whole market. The leader is Illumina. And they have benchtop models. They're relatively small devices. But again, they're still relatively slow for larger volumes of reads.
OK, now you mentioned Moore's law. When is this going to work? When am I going to see this? When am I going to be using it in my data
Well, as I said, we are lining up pilot users. Our machine can do at its roughly 10 or 12 megabits once it's fully optimized. We can do between 100 and 125 gigabytes per day, per 24-hour period. So we have pilot users that are beginning in early 2020 to start to work with us, get a sense of the technology, help us inform our development roadmap.
So I won't be putting
NASCAR
my library,
could theoretically, like I've been bugging you,
there.
sign up
I won't
shortly.
be putting my library in there this year, is what you're saying. So we still have a while before it becomes standard.
Really quick, if you could, sir, because we're out of time.
Paul Steckley, Applied Electronics, so I have to ask, is it alive?
It is not alive, and it's a perfectly reasonable question. It doesn't
Somebody's gonna put it in a
mutate.
frog's egg.
I will say the PCR technique can lead to replication errors, but again, the multitudinousness of the media and the redundancy of it helps with that. But in its static state, and there are kind of preserving chemicals that we can use to keep it shelf stable for even, again, thousands of years potentially, it doesn't really change of its own volition. And I'm sorry, the other part of your question, just does it,
I just
is
said,
it alive?
eventually somebody will put it in a frog's egg.
So it's not to be like inserted into an organism. We're not splicing it in with CRISPR. It is an organic set of chemicals, but it's not a living system.
OK, of course, I'll be there,
Thank
Nick.
you, everybody.
Thank you so much. Let's hear it for Nick Gold.